In this video, we're going to learn about the angiosperm life cycle. And angiosperms, like all plants, exhibit the alternation of generations. So as we mentioned, the angiosperm life cycle does follow the alternation of generations pattern. And like most plants, except for the non-vascular seed plants, most plants have the sporophyte generation as the more dominant generation. And more dominant just means that it's the kind of the more noticeable part of the plant or more noticeable generation of the plant. And the sporophyte generation is the diploid generation. Remember, diploid is 2N, and this just means that the cells have two sets of chromosomes. One set comes from the maternal gamete, which is the egg, and the other comes from the paternal gamete, which is the sperm. Angiosperms are heterosporous, meaning that they have separate male and female structures involved in reproduction. So let's start by talking about the female angiosperm structures first. And we're going to start by doing a review of the structures that you learned about in the video on flowers. So let's take a look at this picture over here on the right. And so this green thing is the gynoecium or gynoecium, which is also just kind of technically referred to as the carpel. And remember that the carpel is made up of multiple different structures. So you have the stigma at the end here. You have this connecting point between the stigma and the ovary, and this is the style. And then this area down here is kind of altogether known as the ovary. And within the ovary, we have these ovules. And these ovules are the megasporangia. So megasporangia. So a megasporangium consists of a single ovule plus some surrounding tissue. So technically it's the ovule plus some other stuff, but we're just going to refer to the ovule as the megasporangium. And within this ovule, within this megasporangium, there are these other cells called megasporocytes. And these megasporocytes are going to be the cells that are ultimately going to turn into the gametes. So these are cells that ultimately are going to form gametes and some other cells by going through meiosis. So if we look at this next bullet point, so megasporocytes, so these cells that we just talked about up here, they are diploid, which means that we also refer to them as 2N. And remember, this just means that there are two sets of chromosomes in these cells. Now, these megasporocytes go through meiosis. And remember, with meiosis, we take cells that are 2N and we convert them to cells that are haploid or 1N. So this one diploid megasporocyte ultimately goes through meiosis and forms 4 haploid megaspores. And you can see that happening over here. So the megasporocyte is up here in the ovaries. And then it's going to go through meiosis to form megaspores. So we can see here there are 4 megaspores in this structure right here. Now, out of these 4 megaspores, 3 of them are not going to survive. Only one of them is going to survive to move on to the next step of reproduction. And I accidentally put this picture on the wrong slide, so ignore it for now, and we'll talk about this on the next slide. So that one surviving megaspore, so the one that we talked about on the previous slide, is going to go through mitosis 3 times. So interestingly, this mitosis is going to result in the formation of 8 haploid nuclei, but we're only going to end up with 7 cells. So one of these cells is going to end up with 2 haploid nuclei in it. So essentially, we're going to start with this megaspore that has a haploid nucleus inside of it. It's going to go through mitosis. And it's actually going to go through mitosis 3 times. And each time that the number of nuclei is going to double. So after the first mitosis, we're going to end up with 2 nuclei. Then we end up with 4 nuclei. And then we ultimately end up with 8 nuclei that are divided amongst 7 cells. And we can actually see those 7 cells over here in this picture. And I know it's small and a little hard to see, but there are 3 cells right here, 3 cells here, and then 1 cell in the middle. So essentially what we end up with is this ovule that is going to have 3 antipodal cells on one axis or one end, an egg cell with 2 synergids, and then a center cell that is going to have 2 nuclei in it. So each one of these cells has a nucleus, and then the one in the middle has 2 nuclei. So to try to make this a little bit clearer, let me switch to some different colors here. So this structure here is the ovule. Each one of these is a cell. Then each one of these cells has a haploid nucleus inside, except for this center cell, which has 2 haploid nuclei. So now let's talk about the male structures associated with angiosperms. So in angiosperms, first of all, let's do a quick review of the structure that we're looking at here. So this is going to be the androecium, which we can also just call the stamens. And each one of these consists of a anther, which is kind of this whole structure here. We have a filament, and then there are pollen grains inside. Now, technically, they're not pollen grains until they get to this kind of part right here. So this is a pollen grain. And this is a single pollen grain. But we're going to talk about essentially how we get from point A to point B by talking a little bit more about these structures. So the pollen sac, which is also called the anther sac, is the microsporangia or the microsporangium, and it is located in the anther of the androecium. So it's inside this anther here. There are diploid, so 2N. Remember, 2N means that there is one set of chromosomes from the maternal gamete that made this plant, and one set of chromosomes from the paternal gamete that made this plant. These sporocytes are just cells that go through meiosis. And remember, with meiosis, we take something that is diploid and we reduce it to haploid. And we end up producing 4 haploid microspores. Now, each of these microspores is going to go through mitosis. And so remember, they are haploid. And so all the cells produced during mitosis are also going to be haploid. And what we ultimately end up with is a pollen grain. And this pollen grain is the male gametophyte. So if we look at this picture over here, so we're starting with our microsporocyte here. That's going through meiosis to ultimately form 4 haploid microspores. And you can see them inside here. So they start out kind of stuck together. So there are 4 of them. Now, each one of these microspores, so if we were to take just one of them, and that's going to show us what is happening here. So this is one microspore that is then going to go through mitosis, and it's ultimately going to form 2 cells that are surrounded by a pollen coat. So you can see the kind of spiky bits here. That's the pollen coat. And then inside this single grain of pollen, we have 2 cells. So it's showing one here and one here. And each one of them has a haploid nucleus. So to kind of recap that, so let's take a look at a microspore. Remember, a microspore has a haploid nucleus in it. And this is that microspore right there. Now it's going to go through mitosis to form ultimately 2 cells with a haploid nucleus inside. And I'm going to draw these nuclei a little different from each other. And that's just to help us distinguish between the 2, because one of them is going to do something different coming up here. Now these cells are surrounded by a pollen coat, which I'm just kind of drawing this like squiggly marks here. So this is just the pollen coat. In some pollen, it is spiky like that. In other pollen, it's smooth. It just kind of depends on what species of plant it is. Now, one of these cells is a pollen tube cell. And the other one is a generative cell. And so this one is actually going to go through mitosis again. So we're going to have our pollen tube cell, which is just hanging out. But then our other cell is going to go through mitosis again. Once again, still surrounded by that pollen coat. And so we're ultimately going to end up with one pollen tube cell. And then these 2 are both sperm. And we'll talk about what they do when we get to pollination and fertilization. Now, these pollen grains are released when the anther dehisces. And dehisces just means that the anther breaks apart. So it might dry out or something, and then it kind of pops open, and then all of the pollen is then released. So now let's take a look at pollination. And one thing to keep in mind is that pollination and fertilization are 2 different things. All right, so do not get these two confused. We're going to talk about what happens during pollination, and then we'll talk about what happens during fertilization. So pollination has to happen before fertilization can happen. And so what happens is, remember that in the female structures, we have this sticky stigma up at the top here. And the sperm stick to that sticky stigma. Now remember that we have essentially 3 cells inside this pollen grain. And some sources call them cells, some just call them nuclei because they're kind of all the same cell. Don't get too caught up in the details. Basically, we have 3 nuclei involved here. So we have the tube nucleus, or the tube cell, which is going to form this pollen tube. And this pollen tube basically just forms a pathway down through the style to get to the ovary. And ultimately, this pollen tube is going to kind of form a pathway through the micropyle. And so if we look a little bit closer here, so if we look closer at one of these ovules here, the micropyle is just this this kind of opening at the base of the ovule. Once the pollen tube makes its way to the ovule, the 2 generative sperm cells are going to be deposited into the embryo sac. And so this whole structure here is now referred to as the embryo sac. Now remember, in the embryo sac, this was produced, this is the female structure that was produced. Remember that it ends up having 7 cells in it. We have the antipodal cells. We have the central cell. And this one has 2 nuclei in it. Then we have the egg, and then the synergids. Now remember that the pollen grain consisted of that one tube cell, but then we also had 2 sperm cells. So we actually end up with something called double fertilization occurring. So both of those sperm cells are going to be involved in fertilizing this embryo sac in a certain way. So one of the sperm cells is going to merge with this central cell. Remember that this central cell had 2 haploid nuclei. And then the sperm cell is also going to add a haploid nucleus. So we're going to have 2 haploid nuclei already there, plus a haploid nucleus from the sperm. And what we ultimately end up with is a triploid cell. And what this means is that this cell has 3 sets of chromosomes. Now, this triploid cell is going to go through mitosis. Remember, mitosis is just producing more of the same cells. And ultimately, this structure, so this central cell with this haploid sperm fertilizing it, is going to create what we call the endosperm. And the endosperm is ultimately what is going to nourish the developing embryo inside the seed. And so these cells are going to produce carbohydrates, proteins, and lipids, which the cells in the seed are going to be able to use to make energy and other structures as they kind of grow and develop. Now, the other sperm cell from the pollen grain is going to fuse with the haploid egg, because remember, in the embryo sac of the female structures, remember we had those 3 antipodal cells, that central cell, which has now turned into the endosperm, and then we have an egg with 2 synergids. So this cell right here is the egg cell. And the egg cell is haploid, so it's got 1 N. And it is going to fuse with the other haploid sperm in order to form a diploid, so a 2N. So now we've got those 2 pairs of chromosomes because we've got one from the egg and one from the sperm. And this is going to form a single-celled zygote. So a zygote is just a single cell. Now that zygote is going to go through mitosis, and since the zygote is diploid, all of the cells produced during mitosis are also going to be diploid. And so ultimately what this forms is a developing embryo. And so an embryo is just a group of cells that typically have not started differentiating, or maybe have started differentiating a little bit in order to ultimately develop into a full-sized organism. So within this embryo, there is a radicle that develops, which we can see right here, which is a small root. There will be 1 or 2 cotyledons, which we will talk about that a little bit more when we talk about monocots and dicots. But essentially, these are just passageways for nutrients from the endosperm to the seed in the embryo. And so you can see those kind of here. And then the seed coat, which makes up the outside kind of protective layer of the seed. So let's review those seed structures again. So first of all, we've got that seed coat, which is just a tough protective outer layer. Integument just means a covering. Humans have skin, and skin is part of the integumentary system. And so our skin is just known as the integument, which is just the covering of our body. The endosperm is going to have those food reserves, and so it's going to be making the carbohydrates and proteins and lipids and providing those to the cells in the developing embryo. And then the embryo is protected and nourished by the seed coat in the endosperm. Remember, this embryo is just going through mitosis in order to make more and more cells. And so we start out with just a couple cells in the embryo. That number doubles. Eventually we have 16 cells, and then 32, 64, et cetera, until we end up with a flower. Now, germination refers to the seed actually growing into a plant. So once the seed has been produced, it's going to be released or carried somewhere else by maybe an animal or water or the wind. And if it lands in a suitable environment, it's going to start going through mitosis. And so remember that embryo, which is inside the seed, is just going to be going through the cell division over and over and over again until we ultimately end up with a diploid sporophyte. And remember, the sporophyte is just the main part of the flower that you're seeing. And then the gametophytes, remember the gametophytes are just these kind of the other alternation of the generation. So the flower itself is the sporophyte. And these cells are all diploid. And then in the middle, or in the center of the flower, remember we talked about the carpels, which are the female structures, and they contain the female gametophytes. So they're not the female gametophytes, but they contain the female gametophytes. And the female gametophyte is the embryo sac. And then the stamen, also in kind of the center of the flower here. These are the male structures that contain the male gametophytes, which are the pollen grains. And then the cycle just starts all over again. So germination produces the sporophyte, which then allows for the production of the gametophytes, which then allows for the production of another sporophyte, which produces more gametophytes, and on and on and on.